Good afternoon, everyone, and thanks very much to the organizers for inviting me here to contribute to this extremely interesting meeting today. So I'll speak about nanobiology for regenerative medicine. We work at the University of Turin, as many of you, at this department, the Department of Clinical and Biological Sciences, that belongs to the School of Medicine. And let me acknowledge, since the beginning, this very young and very enthusiastic group I have, mainly composed of PhD students and master degree students. And one of our main interests in the lab is regenerative medicine, the field that utilizes different enabling technologies like stem cells, nanotechnologies, advanced materials, in order to try to repair damaged tissues or damaged organs. And, well, actually, one of the most important needs in the field of regenerative medicine already a few years ago was that of finding a non-invasive method to track stem cells. And that's why a few years ago we started a very fruitful collaboration with the group of Gianmario Martra, who developed these fluorescent silica nanoparticles you have already heard about a lot today. And we demonstrated that these silica nanoparticles enter very beautifully through endocytosis in different stem cells. Here's an example with human mesenchymal stem cells, but it works with many other types of stem cells. This is a TEM image showing the nanoparticles entering the cells through endocytosis, and this is a fluorescent image showing that inside the cells, the nanoparticle will accumulate into lysosomes. And this occurs in leaving an unaltered cell survival and proliferation capability in a complete absence of cytotoxicity and genotoxicity, and by leaving an unaltered cell stemness profile, and by leaving an unaltered differentiation potential of these cells. So they can be used actually for stem cell imaging. But then we became even more interested into these nanoparticles when we saw that these silica nanoparticles actively engage with human stem cells in improving functional cardiac integration. And we are very interested in cardiac regenerative medicine in our lab. And this was essentially due to the work of Yasmin and Emanuela in the lab. And actually, for a stem cell to integrate in an injured heart tissue, many things are required, especially chemotaxis, in order for the stem cell to arrive to the injured tissue. Then the stem cell needs to adhere to the injured tissue and then start communicating with the surrounding cells, and in particular with cardiomyocytes. And actually, different types of stem cells, including human mesenchymal stem cells, can have advantages for cardiac regenerative medicine, essentially due to their paracrine activity, but actually they still suffer from severe limitations, essentially a very low engraftment ability, a very low survival rate at the site of injection, and a very low interaction capability with surrounding cells. So acting on these three problems will greatly improve our capacity to address this issue. So we started from chemotaxis, and we observed what happened to the stem cells which had internalized the silica nanoparticles. And what we observed was that they can now better express on the cell surface in a statistically significant difference between treated cells and control cells, a very important chemokine receptor, which is CXCR4. And this is very relevant. The axis CXCR4, SDF1 alpha, which is its ligand, is very, very relevant in the cardiac field. And thanks to this increased expression, then now these nanoparticle-treated cells are more capable of migrating, of performing chemotaxis against SDF1 alpha. These are transwell chemotactic assays. Then we looked at the cell substrate adhesion of the treated cells, and we were very interested into the focal adhesion complexes that are anchoring the cells to the extracellular matrix. And what we observed was that silicon nanoparticle internalization can actually modify the adhesion complex structures that now become more elongated. And this difference in elongation is statistically significant, and thanks to these more elongated, more mature focal adhesion complexes, now these cells are more capable of adhering to different extracellular matrices. This is an adhesion assay on fibronectin, this is a detachment assay from laminin. And then we looked at the intercellular communication, and we are very interested into the gap junction channels, which are these important channels, especially those composed of connexin 43, are very, very relevant in intercellular communication at the myocardial level, and also the electrical coupling of the cells depend on these channels. And what we observed was that silicon nanoparticle internalization can modulate gap junction protein expression that now become more expressed. Not only that, but these gap junction are indeed functional. This is a dye transfer assay, and the mesenchymal stem cells having internalized the nanoparticles are now more prone to transfer the dye, which is a gap junction permeant dye, to recipient cardiomyocytes. So then we wanted to assess whether these interesting, from our point of view, properties, cellular properties induced by the silicon nanoparticles could be maintained in a dynamic condition, mimicking the shear stress forces that are found in the myocardium. So we started this collaboration with Anna Marsano at the University of Basel, thanks to our fellow, Deborah, who started working with this bioreactor. And this is the experimental setting. They have this custom-made shear stress chamber with an oscillating flow, and they mimic these myocardial shear forces that have been mathematically modelized a few years ago to correspond to 0.43 dyne per square centimeter. And so they applied these forces to the cells. And what they observed was that actually these major capability of the treated cells to adhere to the substrate of fibronectin in this experiment was maintained also in dynamic conditions, again thanks to the fact that these focal adhesion complexes elongated a lot. And also the same was true when instead of fibronectin they had a monolayer of cardiac cells. And again, the difference between the capacity of adhering of the treated cells with respect to the control was statistically significant. Not only that, but also in dynamic conditions, the treated cells, nanoparticle treated cells, were more capable now of putting in contact with surrounding cardiomyocytes, thanks again to gap junctions, and connexin 43 expression again increased. So also in dynamic condition we have the same effects. And then we wanted to see what would have happened ex vivo in this isolated beating rat heart. This is a model in which we can induce ischemia through the ligature of the left anterior descending artery vessel, and then we can, this is the infarcted area, and then we can inject either control or treated nanoparticle treated cells into the apex of the heart. And what we observed was that in this model, the mesenchymal stem cells treated with nanoparticles were more prone to engraft into the ischemic area. But what's the mechanism that can explain all these observed phenotypes? What we think is that the majority of the explanation stays in a transient blockage of the autophagic flux that is induced by our nanoparticles. First, we saw that when the lysosomes of cells are full of our nanoparticles, now it is not possible anymore for the autophagosomes that here are marked in red with LC3 probe, they do not fuse anymore with lysosomes. And this is a Pearson correlation analysis showing this lack of co-localization. And here, and second, we observe that if we use a pharmacological inhibitor of the lysosomal degradation activity that acts exactly in this pathway, so in inhibitor of the autophagic flux, we mimic in control cells the same phenotypes of increased cell adhesion, increased connexin 43 expression that we had induced with nanoparticles previously. And third, we know that this blockage of the autophagic flux is transient. Here's cells having internalized the nanoparticles. These are the lysosomes that, upon a time frame of about 7 to 8 days, become completely normalized, similar to the control, and by the same time frame, we lose our phenotypic effect. So the phenotype of the cell now returns to the baseline. So we think that this transient blockage of the autophagic flux can explain, at least partially, the mechanisms, the phenotypes that we see in our cells. So based on this first part of experiments, we can state that nanoparticles should no longer be viewed as inert agents for biomedical applications, but they can actually play a very active role in mediating biological activities. And some of these activities might ultimately result in augmented stem cell regenerative potential. We also work with bio-nanomaterials, and I wanted to briefly introduce to you also this project. We have been lucky. A few days ago, we won a business competition with this project in Pavid. Pavid has the aim of developing intelligent microfabricated patches able to recruit stem cells and control reperfusion injury process for in vivo myocardial implantation. And its mission is improving the care and quality of life of patients with myocardial infarction. We all know how relevant it is, this unmet medical need. MI is the leading cause of death worldwide, and in Europe, for instance, according to the European Society of Cardiology, 1 in 6 men and 1 in 7 women will die from MI. So this poses a tremendous economic burden to Europe and to the emerging markets. And overall, MI is estimated to cost the European economy almost 60 billion a year, about 1/3 directly related to healthcare costs, another 1/3 linked to productivity losses, and the remainder to the value of indirect care. And it's more or less the same in the emerging markets, where the hospitalization cost of acute MI is really a primary concern. MI is still an unmet medical need because the current therapy, which is based on drugs, coronary artery bypass surgery, or angioplasty procedures, are placed in action. However, none of these can restore the functionality of the damaged myocardial tissue. And in addition to this, while the use of thrombolytics or balloon angioplasty to very rapidly reperfuse the heart following MI has today greatly reduced morbidity and mortality, paradoxically, up to 50% of the damage to the heart following MI is the result of reoxygenation, the so-called reperfusion injury. So IMPAVIT concept is innovative because its rationale is to fix the problem by putting two actions together: in situ regeneration using the cardiac patch through activation of endogenous cells from the one side, and cardioprotection from reperfusion injury using cardioprotective small molecules. And since the association of these two therapeutic actions in one single device was new, it was possible to get an international patent on this that has been already granted. And what's the stage of this project development? We obtained a proof of concept in an ischemic large animal model. For myocardial ischemia, the large animal model, the pig, is the only relevant model, actually. And since less invasive implants could actually increase the target patient population, we have begun working with a minimal invasive closed chest intervention. So briefly, these are all published data. These patches mimic the myocardium structure and biomechanics. Many similarities are found from the morphological, chemical, and mechanical points of view. It is based on a cat-based reproduction of a decellularized myocardium from pig. And these patches are indeed biocompatible, and they are cardioinductive. So this patch allows adhesion and growth of human stem cells, and stem cells cultured on these patches adopt a distribution that mimic the three-dimensional cell alignment that we find in the myocardium. And finally, stem cells cultured on these patches begin their differentiation towards the myocardial lineage. They downregulate stemness markers, begin upregulating the cardiogenic transcription factors, and then later on, especially the precursors, cardiac precursors, they start expressing sarcomeric genes and then proteins. These are also data that we have already published. So then we analyze the implant feasibility in vivo in this ischemic pig model, in which we induced ischemia through lead binding, and then we apply the patch before the reperfusion step. And we demonstrated that the patches are easily implanted. They adhere perfectly on the epicardial surface of a beating heart, and after 24 hours, they are still perfectly adherent on the epicardium with a few inflammatory cells around, and after 3 months, they have been completely reabsorbed, resulting in a non-fibrotic, non-inflammatory outcome in the infarcted area. So, but why should these patches work, succeed, when previous approaches failed? We think that IMPAVIT could provide some solutions to previous problems. For instance, the fact that cardioprotectant are usually given in bolus. And this shows a very high systemic side effect and very low local persistency, while in the case of IMPAVIT, we would increase local bioavailability of the cardioprotectant in the absence of important side effects. Second, because the cellularized patches require very long culture time and dedicated cell factories, and we don't have time in the acute phase of an MI. And while with the IMPAVIT, we should have an immediate applicability of the device without delays to cell culturing, and also with a higher product transportability and storage and a complete feasibility within industrial facilities. And third, open chest surgery is often required for previous approaches, while our goal is to deliver the patch in a minimally invasive way. So we developed these milestones that we will try to address in the next years. This is the IMPAVIT team. This is an interdisciplinary team with a cardiophysiologist, biomedical engineer, and myself, a geneticist. We are organizing our advisory board. We have some clinical staff already there since the beginning of the project. And now we are filing other research grant applications, of course, and we are building and testing prototypes in new and improved prototypes, as well as filing new patent claims. And only a couple of more slides, just to introduce to you this Translational Research Center for Innovative Medicine, which is an initiative of the University of Turin within our department. The ancient laundries of our hospital, University Hospital San Luigi, have been completely restored, and new laboratories are being created. They will be like this by the end of the year. Now they are almost completely finished. We have completed. We have a vision, we have a mission, but what is most important to me today is that among the research fields that will be interested in the Translational Research Center for Innovative Medicine, there's nanomedicine. So, of course, the collaboration of all of you will be extremely important to us. So finally, a big thank to all the groups that have collaborated with us in the two projects that I've presented to you today, and these are the funding sources that are active presently in our lab. And thank you very much for your attention. I'd be happy to take your questions.